Peace be upon you all, and welcome to another episode of the exciting series Seeing God in Biology with Dr. Fuz Rana. And today we are going to talk about a marvelous topic, protein folding. So, first of all, Fuz, how are you doing today?
I'm doing good, Ahmed. As always, it's great to see you and great to spend time with you.
Same here. So protein folding, it's nobody, the layman wouldn't know why proteins need to be folded anyway. So
Yeah,
why are we folding them?
yeah. Well, uh, proteins are very important molecules in biochemical systems. I like to call them the workhorse molecules of life. That virtually every structure in the cell, virtually every activity in the cell, is formed and mediated by a large number of different types of proteins that are working collaboratively. And so even the simplest cell, gosh, must have at minimum, several hundred different types of proteins just to be able to carry out what are considered to be the non-negotiable essential biochemical operations.
Mhm.
For a system to be recognized as a life form, as a true bona fide cell, it has to have, again, minimally, several hundred proteins. And the cells that can live independently on their own in the environment typically have in the neighborhood of somewhere between 1,500 and 2,500 different proteins, depending upon their metabolic lifestyle, whether they're chemoautotrophs or photoautotrophs. So proteins are really critical molecules, and um, they're these long chain-like molecules
Mhm.
that have to fold into complex three-dimensional shapes, and it's the precise shape of that protein, the precise three-dimensional structure, that actually determines its functionality. And if that
Uh-huh.
structure deviates ever slightly from its native state in the cell, that protein will lose function. So the process of going from a linear chain of amino
That's
acids,
strange.
as we talked about in previous episodes, to a precisely folded protein chain is called protein folding.
Protein
And it's
folding,
a
yeah.
it's a and it's one of the most challenging problems right now in biochemistry. Is how does amino acid sequence translate into the three-dimensional structure of proteins, and hence the protein's function.
Yeah, so for the audience, I would really recommend that you would all, of course, watch the whole series from the beginning, but for sure watch episodes 5A, B, C, D, and E, which took us in a journey from starting from DNA and then reading DNA into RNA and then reading RNA into or translating into what has been translated into RNA is then moved into protein, and then how this protein is trimmed, the excision process, and now we have this string of of protein that is right about to form the machine. And then today we are going to see this very strange process. As an engineer, I find it very fascinating that a complete machine is actually just a linear set of things that then just can come together into it's already very fascinating to see origami, for example,
Yeah.
where you start by a piece of paper, which is a 2D object that you can then make into a 3D one. Now it's so amazing to start by a linear object, something that is
Yeah.
actually a line, and make a complete 3D machine out of it. So very, very
Yeah.
fascinated and excited to look at that. So
Yeah, well,
let's go
I've
to the
got some
presentation, maybe.
slides. Yeah,
Yeah.
and so this is a great summary of what you just described. Which is
Yeah.
the central dogma, where, again, as you're saying, DNA contains the information needed to make proteins in a sequence of nucleotides known as a gene
Yeah.
that's copied into RNA, which then is translated at ribosomes into proteins. And this is a slide that kind of shows a cartoon of that process of protein production at the ribosome, where
Hmm.
the ribosomes are like these Turing machines, these biochemical Turing machines that are reading, digital data in the form of messenger RNA and are producing a digital output in the form of protein chains made up of a sequence of 20 different amino acids. And so the question then becomes, how do proteins fold? And that process again is summarized, at least in a conceptual way, with this cartoon that shows again that linear chain of amino acids, where the backbone will fold into typically one or two, broadly speaking, one or two conformations, either an alpha helix or a beta pleated sheet. And then those then will interact with each other, those folded regions, in three-dimensional space to produce the three-dimensional structure of a protein. This is called its tertiary structure. And then many times those folded protein machines will interact with other folded protein machines to form complexes that themselves are machines. So it's almost like you have a modularity, to the design of these massive protein machines. So the question then becomes, how does that folding process, take place? And
so you explained this. Actually, it goes into many steps. So the string becomes a shape, and then completes the shape, and then it can actually even come together with other strings already shaped. So it's a bigger machine, let's say.
Yes. Now what's interesting is that there are some proteins that will spontaneously fold into their three-dimensional native state structure. This is biochemists will refer to a protein when it's properly folded as its native state, and that tends to be at what biochemists also refer to as an energy minima. In other words, the chains fold in such a way so that the overall protein structure has minimal energy. It's at a minimal energy state. And so if you think about protein folding, as kind of a landscape, where different folding options will occupy either valleys or hills, typically the linear chain is occupying in terms of energy kind of the top of a hill, and as it folds, it can either go to a higher hill or go into a valley. But the lowest possible valley would be the energy minima, and would be that folded geometry at that point would be referred to as the native state. And some proteins will spontaneously fold into their native states, but others actually can't do that. They actually require assistance in order for that to happen. And there are a set of proteins that are called chaperones that actually facilitate that process. In fact, some people think that as the protein is exiting the ribosome, that part of the ribosome itself may actually facilitate the protein folding, may function kind of like a chaperone. And so, chaperones are, the people that you send out with your kids when they're on a field trip or what have you, to make sure that they behave.
To take care of them,
Yeah. And so that's what these proteins are doing. And some are low molecular weight proteins. These are called the heat shock proteins that will kind of bind to certain regions of the protein, particularly those regions that are hydrophobic, preventing them from kind of sticking together in an inappropriate way because
they are trying to escape the water, the hydrophobic ones.
Right. And that can lead to the proteins, the chains folding improperly or aggregating with other protein chains to form, this mess that where the proteins can't fold. So you want these chaperones to kind of help prevent that from happening.
So they like go to those sites and like protect them from the water so that they don't rush into collapsing the whole thing.
That's exactly right.
Yeah.
And then there's another group of chaperones that are interesting, and this is characterized by what's called the GRO-L complex that is found in E. coli, but there are other versions of this in other organisms. And these are these massive, this is a massive protein complex that forms a barrel shape. like a barrel where there's an internal cavity that
Mm.
that protein chain will enter into, and
Oh.
it creates kind of this safe space in this interior space that allows the protein to fold. And then when it
Mm.
reaches its proper folded shape, it'll exit the barrel
Ah.
as a properly folded protein.
And does the barrel itself affect the way it folds, or it just protects it from the environment so that it folds? It takes its time, let's say.
Yeah. So the chaperones and the both types of chaperones are not going to alter the native state structure, because that's a thermodynamic state.
Uh-huh.
But what they will do is they will influence the kinetics of the process. So the way
Mm.
chemists think of things is something's either kinetically controlled or thermodynamically controlled.
Mm-hmm.
And so a great example of the difference would be, uh, think of carbon in its elemental form. The
Mm.
lowest energy state of carbon is graphite,
Yeah.
right? So carbon is going to naturally want to uh, go to the state of being
To have safe.
graphite.
Yeah.
Okay, and so that's the thermodynamic minimum for carbon.
Mm. Mm.
But diamonds are pure carbon, or at
Yeah.
least, if it's a pure diamond, it's pure carbon.
Mm-hmm.
And diamonds are actually less thermodynamically stable than graphite. And so, from a thermodynamic standpoint, diamonds should actually convert into graphite. And in fact, they will. But the problem is that process is so slow that, for all intents and purposes, carbon as a diamond is trapped in that state. So
Ah.
that's a kinetically controlled process, because it's so slow that process can't happen in any kind of appreciable time. So
Yeah.
that's the difference. And so with protein folding, the final state is thermodynamically determined, but the pathway to go from that linear chain to that final state is kinetically controlled. And so what you're doing with the chaperones and with the GROAL complex is creating a kinetic pathway that facilitates going to that energy minimum.
Yeah.
Whereas if it wasn't for these, then these hydrophobic regions would interact, and either the protein would improperly fold or would aggregate with other improperly folded proteins and now be kinetically trapped in that state, just like carbon would be trapped in the diamond state. So
Yeah.
that's what's going on from a kind of a thermodynamics and a kinetic standpoint.
From a design perspective, I would like to ask you: is there a reason that chaperones exist in the cell? Do they do anything else, or they are
Well,
there for helping proteins? This is what they
yeah,
do.
and I don't have a ton of expertise on the chaperones,
Mm-hmm.
but the reason why in that diagram you have a chaperone labeled HSP70, it stands
Uh-huh.
for heat shock protein. And the way that people discovered these chaperones was recognizing that when the bacteria E. coli, for example, was put under heat stress, it would actually produce these proteins called
Hmm.
heat shock proteins that would actually bind to proteins, protecting them from essentially unfolding and forming these aggregates. Because when a protein's in its native state, if you heat it, it'll
Hmm.
start to unfold and will expose hydrophobic regions that will then
Ah.
cause the
Yeah.
protein to lose its structure, folded structure, or to aggregate with other proteins. So the heat shock proteins are this protective measure. And so people discovered that not only are they're the heat shock proteins that serve kind of as a stress response, but some of them are actually expressed constitutively uh, as as a way to help proteins fold. So
Hmm.
I'm not sure if that answers your question exactly, but uh,
It does answer it, because what it says is that, chaperones are there to help proteins and to protect proteins. So I mean, they are existentially attached to proteins. So then the question is, what if those guys were not there? What would be the fate of life? I mean,
yeah.
you
Well,
cannot wait for a protein to fold in a million years.
yes, yeah, and what'll happen is without
Yeah.
these chaperones, most of the proteins are not going to fold properly, and that
Mm.
really, as you're bringing up this whole idea of of the design argument, that kind of leads us to,
Yeah.
what I think you're getting at, which is, it's a chicken and egg problem, right? Because
Absolutely.
in order to have proteins, in order to make proteins, you have to have proteins,
Yeah,
right?
absolutely.
Right.
Yeah.
And it's not just simply limited to protein folding. I mean, this problem is much more extensive, because to make the messenger RNA, the transfer RNA, the ribosomal RNA that are critical for the whole process of protein production, this is all mediated by an ensemble of different proteins. You have
Yeah.
to have proteins to attach the amino acid to the transfer RNA molecule. There are proteins that will ferry the charged transfer RNA to ribosomes. You have to have these, elongation factors that are interacting with the ribosomes. The ribosomes themselves consist of proteins, right?
Yeah.
And then with protein folding. So every step of the process of making proteins requires proteins. And so when you have this kind of a system, this is a signature for a design system.
Absolutely.
The only types of systems, I mean, chicken and egg systems are very common. Uh, where
Yeah, where it's definitely, it's showing that there is an external agent who has put together the parts. Otherwise, the parts cannot come from nowhere, because they depend on their pre-existence, let's say.
right. So if you're manufacturing steel,
Yeah.
right, you need to have machinery that's made out of steel in order to manufacture that steel, so to speak,
Yeah.
right?
Yeah.
And so those, to me, chicken and egg systems, you could think of them as a special type of irreducibly complex system,
Absolutely.
but
Yeah.
they're distinct in the sense that, with irreducibly complex systems, it's that you're just saying all of the parts have to be there for that system to have minimal function, right? And they have to interact precisely. With a chicken and egg system, what you're saying is that, that not only all the parts have to be present, but the parts that you need to be present are made up of the parts that you're making, so to
Yeah.
speak.
Absolutely. And the chaperones themselves, they are they also classified as enzymes?
Yes, I think you could reasonably call them enzymes in that sense.
And they are enzymes, protein enzymes, not like RNA components.
Yes, exactly.
Yeah.
They are proteins.
Yeah.
Yes.
So again, amazing. So it's not only that the ribosomes themselves have plenty of protein components, apart from the ribozyme, but actually even for the proteins themselves to eventually survive,
Right.
they need those proteins to help them. And this would put another big question mark on the RNA world hypothesis kind of evolutionary thinking, wouldn't it not?
Yes, it would, exactly.
Yeah.
Because it's hard to envision how you would somehow, from an RNA world, be able to invent a kind of a DNA protein world, uh, because you have to have so many different types of proteins present, in order for that protein world to emerge. It can't emerge in a piecemeal manner, because in a previous episode, I think we talked about the auto-destruct cycle, right? That if ribosomes are made out of proteins, and if those proteins that are part of the ribosome are not made precisely correctly, you're going to wind up with ribosomes that are defective, which means the next round of protein synthesis is going to produce ribosomes that are even more defective, and you
Yeah.
wind up in an auto-destruct cycle. Well,
Yeah.
something comparable is going to be true with chaperones as well. That if those chaperones are not produced precisely as they
Hmm.
need to be produced, they're not going to be able to facilitate protein folding, and
Yeah.
therefore you're going to wind up with
It's a downward spiral, essentially. So
yes, yeah,
bad chaperones, bad proteins, and then bad enzymes, and then it just gets worse, and life will not have a chance. Yeah.
right. And what that is also saying is that you can't go in the reverse direction either, right? You
Hmm.
can't go from a crude system to a perfect system. Uh,
Ah.
because you can't reverse that auto destruct cycle, which is what you would have to do in an RNA world. Right.
Wow, that's a great insight. So if the system doesn't start as a precision system, it only has one way to go, which is downhill.
Yeah.
It needs to start at a precision system that autocorrects, that is really good at what it's doing. Otherwise, it's just going to be, things will just get worse from there. All right, so let's go on with your presentation then.
Yeah. So then now this is where things, I think, become very interesting. Because, again, we're talking about the fact that, we're looking at the three-dimensional structure of proteins. And that really is what's important. And I actually love your origami analogy, because if you have a single, protein chain that's made up of 150 amino acids, right? The estimation is that I've seen is that there's 10 to the 68 ways that could possibly fold. Right. So when you're thinking about protein space, right, it's not just simply with if you have 150 amino acids, you would have 20 to the 150th power possible sequence combinations, which is incredibly vast. But then for each one of those sequence combinations, there's 10 to the 68 ways that you could possibly fold them. Yeah. So you're now looking at 20 to the 150th power, all of that quantity raised to the 68th power. Wow. And so it's an incredibly vast,
huge amount. Yeah.
Vast protein space.
Absolutely.
And so, so the question becomes, how do proteins fold? If there's, if for a single protein chain, there's, again, of 150 amino acids, which is about an average protein, right? That's an average size for a protein, maybe on the small end. There's 10 to the 68 ways it could fold. And this led to what's called the Leventhal paradox, which is, how do you explain how proteins can fold to their native state so rapidly?
Yeah. Right? How does it find the right solution is the
because even if you could explore, one fold per second, or one native state, or one possible folded structure per second, you just don't have enough time. And yet proteins will go boom, on a micro scale, they on a micro time scale, they'll fold. I think it's on the order of, maybe milliseconds that a protein will adopt its native state structure. Wow. And so this is a diagram that biochemists use to kind of illustrate the Leventhal paradox, where the top is at the high energy state, and you have these unfolded chains, and so you'll have these potentially folded intermediates, and we'll get into that in a minute, where you're looking for that kind of that low energy native state. But it's a very precarious situation, because if you don't have chaperones present, you're going to much more likely go into an unfolded or a partially folded or an incorrectly folded state that is going to form these non-functional structures that will just clutter up the internal operations of the cell. Yeah. And so it's doubly precarious in the sense that, how do you find the time to fold? And you don't have the luxury of exploring all the possible spaces, because many of them are going to lead you into no man's land, right?
Yeah, where it's in engineering or in computer, logic, or actually even in mathematics, there is the global minimum or the global optimum and the local optima. And I think the problem here is, how do you guarantee that the protein will not fold into a local optimum and just be trapped there until some event causes it to destabilize and change its shape, which will create chaos, right?
Yes, exactly. Yeah. So this is, and so it turns out that the solution to the Leventhal paradox has to do with the idea that the protein chain kind of folds locally. And so, you've got this long protein chain, but what ends up happening is that along the chain, you can kind of envision these local environments that will fold to minimum states. And then those folded minimal states will then again fold into other minimal states kind of in a sequential manner. And so you're not looking to explore everything, but the folding is constrained by these local environmental effects that ultimately lead you to that energy minimum in the native state, where again the chaperones can help facilitate that process. And whether our viewers realize it or not, we actually talked about this concept when we talked about why the 20 amino acids. Because remember, we talked about the unusual nature of the amide bond, right?
Yeah.
And when we talked about the unusual nature of the amide bond, sorry, I'm jumping ahead. Let me just, sorry, I got the slides out of order here. Oh, you know what? I lost those slides.
Oh.
Okay, so never mind. It's all right. My bad. But I meant to put the slides in, and somehow I didn't do that. So
maybe they are hidden. Maybe they are hidden in the presentation.
Yeah, well, anyway. If people go back to episode 4, we talked about the fact that the amide bond actually is this highly unusual bond that only allows, for geometric reasons, only allows the backbone of the protein to fold into either an alpha helix or a beta pleated sheet. That there's a number of constraints, and so it's those very physical constraints that are actually contributing to this idea of local folding, where it's very easy for a region of the protein chain that may be, that may be 10, 15 amino acids in length to very quickly adopt an alpha helix or a beta pleated sheet. Right. And then it's very easy for those to interact with each other, right, to form, higher order folded states.
Yeah.
So,
so the basic building blocks of the peptide chain is selected to the extent that this local folding can accelerate the process, because distinct parts of the chain will make those plates or helixes that then can now become a bigger building block to form the final shape.
Right, exactly. And it turns out that biochemists have discovered that even though we talk about this massive amount of protein space, 20 to the 150th power, all of that raised to the 68th power, as it turns out that in nature, there's only a few thousand distinct ways that protein chains will fold.
Wow.
Right. And it turns out that each of these folds actually can carry out a wide range of functionality. So these are very versatile folds, and different amino acid sequences will actually yield the same fold. And all of this suggests that it's not just only the formation of the alpha helix and the beta pleated sheet, but it's the way that those interact that seem to be constrained by the laws of physics. There's some kind of physical chemical constraints that limit the way that protein chains can fold. And for example, biochemists have discovered that, for example, alpha helices can only interact with other alpha helices in very specific ways. Two of those are called the knob and hole and the groove and ridge interactions. And so it's, you have these sets of concentric sets of local folding events that are happening that are producing the native state structure.
Yeah.
And there's a famous
who could have thought of it?
Huh?
Who could have thought of it that those amino acids can do that? So it's a huge amount of forward sight, let's say.
Oh yeah, it's incredible, because it's as if built into the very laws of nature are these constraints that are leading proteins to fold in precisely the way they need to fold to be functional molecules. Because you could envision, there might be constraints, but those constraints could be such that they produce folds that are completely useless for living systems.
Absolutely.
So it's this eerie coincidence that the laws of nature are giving you these precise folds that produce functionality, but also these create this capacity for this folding to happen kind of in localized environments in such a way that it leads to this native state that is functional. So there's a famous protein chemist who said that helices and sheets packed together in one of a small number of ways, the connections between secondary structures obey a set of topological rules, right?
Yeah.
And so again, this is, to me, it's a bit eerie. And one of the most interesting studies I've ever come across when it comes to protein folding, which really to me raises all kinds of questions about the fundamental design of living systems, about kind of the teleology of life, is it was done by these two biophysicists who did a thought experiment. And they said, let's assume that you can create a tube, a flexible tube, and that tube is made up of tethering together discs, where in their model, each disc represented an amino acid. So you can envision kind of tethering together these discs of amino acids that then can fold. They're flexible chains. And the 20 different discs that are used have different physical properties. They're either water soluble or water insoluble. And that chain then, of course, is going to want to interact with itself in such a way to kind of squeeze out any water from the interior for those regions where you've got hydrophobic discs, right? Right. So essentially, they're constructing kind of an analog to a protein, right?
Yeah.
With their model. Well, then when they explore, and then they come up with this numerical quantity called a folding parameter, which is the ratio of the tube diameter to the range of attractive forces along the chain, right? So the chain it has a diameter and then it can fold to interact with itself. So that ratio is the folding parameter. Very simple concept. And so if the folding parameter is much greater than 1, the tube is too fat to fold, right? And if it's very much smaller than 1, it's going to be like a string, which means there's an infinite number of ways to fold. But when the folding parameter is 1, you get this balance between, the chain resisting folding and being able to fold. And
the chain cannot fold in very small intervals, because then the diameter will constrain it. Right. So you can only, if it's that long, then there can only be those amounts of interactions, because it will actually essentially be segmented to an extent in relation to its diameter. Yeah.
And so they discovered that when you're looking at a folding parameter of 1, you get about a few thousand folds that are exactly identical to the folds that you see in proteins.
Wow.
And it turns out, guess what the folding parameter is for proteins?
Not 1.
It's 1.
Amazing.
It's so what's crazy here, Ahmed, is that think about this. You've got proteins made up of amino acids that link together to form these protein chains, and the average geometry of that chain, or the average diameter, is and the distance that the chain can interact with itself gives you a folding parameter of 1, right?
1. Which
then in turn means that just simply through geometric considerations, the geometry of the chain and the hydrophobic effect, trying to avoid hydrophobic residues, trying to avoid water, those two parameters lead to the folds that you see that occur naturally in nature, right? That occur in proteins, and they again happen to be exactly the folds that you need to have this versatile range of functionality that you see in proteins.
Amazing.
And so it's like there's this
it's conspiracy.
Yeah.
So the same word. Yeah, it's a
So it's
conspiracy,
like,
and it's working bottom up to reach this goal in from the properties of quantum mechanics to the chemical properties to the electrical properties of everything, and then it boils down to geometry at the end of the day, which is
right.
which is mathematics. So
Yeah.
it's a
Yeah. So
mathematics
there's,
in a mind, essentially. Yeah.
yes. Yeah. So there's this mathematical elegance to the way proteins fold. But this again has some very interesting implications, because
Hmm.
when you have a limited number of possible folds, now instead of trying to explore 10 to the 68 folds, you're looking to explore 10 to the 3 folds, even if
Wow.
it was just totally random,
Yeah.
right? So now the folding problem becomes completely tractable. But when you
Absolutely.
couple that with the idea of local folding environments that
Yeah.
are only going to be able to interact and fold in certain ways, you have now a system that can very rapidly find its native state. But it's because it's been designed, you know, with respect to the very fundamental laws of nature, so that,
It
it
finds the
actually will
solution.
almost spontaneously adopt the shape that it's supposed to adopt, which
Wow.
which to me is, just
It's
mind
completely
blowing.
mind blowing. Absolutely. And when I see those, computer rendered graphics, and the protein is like, it's like shaking.
Yeah.
What's the role that water has to do with it? This shaking action that maybe is giving it the possibility to try to, as rapidly as possible, try the different permutations
Yeah.
and reach to the final solution. It's one of those funny properties of water, isn't it?
Yeah. I mean, it would be the same kind of forces that would cause small molecules in water to diffuse. It's just
Yeah.
that the chain is made up of small molecules linked together. So that the chain structure prevents that diffusion, but those individual components are still trying to diffuse. And so it creates these molecular dynamics that
Yeah.
are definitely contributing to the folding process.
Yeah, and the property of being hydrophobic or hydrophilic, this mix that also causes the protein to, a part of it is trying to escape the water, another part of it is running after it. So it's automatically creating this jitter and wiggling
Right.
effect that essentially the right pieces will eventually find each other in a way, with the help of the chaperones and the protection that's taken place.
Yes,
it's
exactly.
it's it's amazing. I'm trying to think, if humans are to engineer something like this, it needs more than a super genius. I don't think this is can even be something that any human can invent.
Yes, yeah,
Yeah.
it's amazing. Well,
Yeah.
and this is an interesting statement from the two
Mm,
biophysicists that did this study. And they said, the common belief is that given a sequence of amino acids with
mm,
all the attendant details of the side chains and surrounding water, one obtains a funnel-like landscape with the minimum corresponding to its native state structure.
mm,
Each protein is characterized by its own landscape. In this scenario, the protein sequence is all important, and the protein folding problem needs to be attacked on a protein by protein basis.
mm,
They say, but protein folds do not evolve.
mm,
Rather, the menu of possible folds is determined by physical law. In that
mm,
sense, it's as if evolution acts in the theater of life and shapes sequences and functionalities, but does so within the fixed backdrop of Platonic folds. In
mm,
contrast, our model calculations show that the number of common globular proteins reflects a deeper underlying unity in their behavior. This landscape is pre-sculpted by general considerations of geometry and symmetry.
wow.
So, of course, these guys are looking at this problem from within the framework of an evolutionary model, but what they're saying is absolutely profound, because what they're saying here is that the folds that we see in nature are not folds that were shaped through evolutionary processes, through a historically contingent process under the influence of natural selection, but
Wow.
rather it's the laws of physics that fundamentally say these are the only possible folds that can exist, and they turn out to be these just right folds. Right?
Yeah. Actually, just this third paragraph I would say, is enough to eliminate an unguided anything. It just
Right.
says that the one who has created this universe, or who has actually set the basic parameters of, or the basic constants of physics, intended that life is possible. This is a universe which is designed for life, let's say.
Yes.
Because this and the intricacy and amazing things that those machines can do. I just the rendering of those walkers, the kinesins, for example,
Yeah.
it's just completely amazing to imagine that anything like this can be by a strike of luck or an evolutionary process whatsoever. The between brackets pre-sculpted thing is essentially foresight, and nature doesn't have any foresight, I would say.
But you know, and this is a point that you made earlier, but I think, in my opinion, it bears repeating, because
Yeah,
I mean, in a sense, what you see here is that, first of all, you have these fundamental constants of physics that are just right so that life can exist in the universe.
yeah,
And then those fundamental constants of physics create molecules like carbon that has this unique set of properties that make life possible, that gives you the chemical diversity you need for living systems. Those
yeah,
same laws produce water that has these just right, highly unusual, but just right properties that create a solvent system that, working in conjunction with carbon, creates this environment so that life
absolutely.
can be possible. And then out of that, you have these biomolecules that have these
Yeah,
just right properties, so that the only thing that they can do is form these systems that are highly conducive and necessary for life. So it's like you've got
absolutely.
this stacked set of coincidences, right?
Yeah,
That,
yeah,
that
it's
you know, that the emergent properties that come out of one level of complexity as you move to the next regime of complexity, those emergent properties are producing these
yeah.
anthropic coincidences at every
and
level.
and then the signature to find in the core of this information that is embedded in DNA that will pick those, actually not only the information, the very existence of DNA and then RNA, and then the selection of the 20 amino acids specifically with their specific properties, and to the extent that then those laws of physics can be actuated,
Yes.
because you are exactly picking the materials that will put into action those prescribed laws of physics that will cause those pre-sculpted machines to be sculpted eventually. So it's totally. I would say that by any whim of imagination, thinking that this can actually be anything like a coincidence is totally absurd, I would say.
Well,
And it just drew my attention now. This Banavar and Martin in Fitness of the Cosmos for Life actually is a very is a very good title.
yes, yes,
Yeah,
yes. Well,
yeah,
you know, and what's, you know, to kind of build off of your point too, is that these proteins, when they again form these folded chains, will then interact with other proteins, and actually there's a set of rules for how protein subunits interact, but we won't get into that. But
wow,
that's, you know, in and of itself mind blowing. But then
yeah,
they will form these structures like kinesin
yeah,
or ATP synthase, which are like these rotary motors that have this eerie resemblance to the machines that we would build,
yeah,
right? You know,
absolutely.
so, you know, it's,
It's
or
completely
that you see
amazing.
these chicken and egg systems, right? So there's
Yeah,
all these other signatures for design that you see on top of this, that the laws of nature are producing these systems
absolutely.
that, you know, where again it looks like this conspiracy, but then the conspiracy is such that there are these screaming or these, you know, beacons for design and
Yeah,
information, and then the machine like nature of these systems, or the chicken and egg nature. So it's
absolutely.
it it's design all the way down, I guess.
Absolutely, absolutely. Right, great. So that was a very insightful quotation, amazingly from people who are evolutionary in their thinking. But at the end, truth prevails, doesn't it? So
Yeah.
you write evolution, and then you say, "Wow, that." that doesn't look like it. So
Yeah.
once again, this is really amazing. But maybe the last piece that you've just mentioned is that even the protein machines, the workers of the cell, are typically not just one protein that has folded. So things like hemoglobin, for example, are many parts. Ribosomes have many parts. Kinesins and dyneins, etc. They all have many parts, and it's amazing that those parts would just form by different transcription processes, and then they find each other and they come together, guided somehow through other mechanisms in the cell. And then here you go, you have the machine. It's in the right place in the right time. It's doing its function, and everything is in harmony. Well, God is great, I have to say. So there is nothing else. So in Arabic we say Subhanallah. It means like God is exalted, is God, or it's like God is great. And this is really seeing God in biology, I guess.
Yeah. Well, I think your response is very appropriate, because if you don't have that sense of worship, right? In awe, one of my favorite psalms is written by David. It's Psalm 19, where he talks about the heavens declaring the glory of God.
Absolutely.
And then he also reflects upon the law, and his ultimate response to seeing God's glory in heaven and seeing this perfect law from God is, may the words, or how does he say it? May the words of my heart, and may the meditations of my heart and the words of my mouth be pleasing to you. So his response to God's grandeur is essentially to fall in humble worship before God. So that we may all do that.
May we all do that. And that was really a great, I guess, sneak peek into protein folding, which I'm sure is huge, and there are much more details, and even the bigger machines coming from subunits, etc. But I think this can open up for plenty of the audience to do further research about that, and might also spur a multitude of questions. So if you have questions, just push them to us in the comment section, and we might as well just collect them together and do an after show at a later stage, where we'll be collecting enough questions to do that. So please do, and please don't forget to like and share this to the ends of the earth, and to glorify God and say, with your heart, all the words, and use this knowledge of science really to, guess what, see God in biology, metaphorically at least. So thanks a lot, Fuz. This has been as exciting as always. And for everybody, thank you for being here, and looking forward to your contributions. And please be with us next week, where we'll go into the next exciting installment of this series. Which part are we going to be talking about next?
The genetic code.
Yeah, the genetic code. So let's look about a very interesting topic, because people who don't believe in an overarching mind would just hate you for saying code or program or this kind of thing, because it immediately says that there's an intelligence, which we all know that there is. So thanks a lot, Faz, and God bless you, and God bless the science that comes to bring us closer to God, and glorify Him in our hearts, and with our tongues, and hopefully worship Him in our life until we meet Him. So thanks a lot, and see you again soon.